Thanks to the organizers for inviting me. I'm actually not going to, I don't know why they invited me, because I'm not going to say anything about the brain or neural function, but I will give you an introduction to the biology of human L1 and other retrotransposons, and talk about their potential role in tumorigenesis. So retrotransposition is the process by which a region of the genome, an element, is transcribed into RNA. This RNA is reverse transcribed and integrated into a new site in the genome. So this is a duplicative process. We go from one copy to two copies. And here are the transposons that are found in human DNA, many of which are inactive. We have DNA transposons. These are cut and paste elements as contrasted with the retrotransposons. And they contain a transposase to get them to move from one site to another. They make up about 3% of the human genome, and they're all inactive. Okay, there are active ones in bacteria and fruit flies, but we don't have any that are active. They may well have been active millions of years ago in the human lineage. For retrotransposons, we've got a couple of kinds of autonomous ones. We've got long-terminal repeat, or LTR, retrotransposons, which look a little like retroviruses. They've got GAG, pull, and envelope genes. These are endogenous retroviruses. They're stuck in our genome because they have a lousy envelope gene. They can't go from one cell to another cell. They're stuck in the cell in which they're made. And they make up about 8% of the human genome. And as far as we know, there have none that have been found to be active in present-day humans. Okay? We've got so-called line elements, and these are the elements that are active today. And we'll talk quite a bit about them. And they have endonuclease and reverse transcriptase activities, and they drive the expression and retrotransposition of ALU elements, which are about 1 million or so in the genome and can't encode protein. Also SVA elements, which are these composite elements found in about 3,000 copies in the human genome, and also can be reverse transcribed and integrated. And processed pseudogenes, which are messenger RNAs that have lost their introns and can be reintegrated into the genome. So here is the percentage of the genome, which is these transposable elements, almost all of which are dead. And you can see a whole bunch of different mammals. Here's humans down here, about 45% of the human genome being these almost all dead transposable elements, lines, signs, LTR elements, the endogenous retroviruses, and DNA transposons. And you can see mouse, rat, et cetera. They're all in the range of 40% or so of the genome. So how do we study retrotransposon biology? Well, we can isolate and characterize recent disease-producing insertions in humans, and we've done that, as you'll see. You can use in vitro biochemistry to study L1 encoded proteins and the L1 RNA protein complexes, the so-called L1 ribonucleoprotein complexes. You can use engineered L1s and ALUs, SVAs, and processed pseudogenes driven by L1s to analyze retrotransposition in cultured human cells and in transgenic mice. You can analyze whole genome sequences in databases, and there's been a lot of this going on in comparative genomics. And you can now find all recent L1 insertions in any genome by high throughput sequencing, and I'll talk about that in a bit. This is the young man that got me into this field. Actually, it was in the mid to late 1980s. And we were studying 240 males that had hemophilia A, the X-linked disorder that Andy mentioned in his talk. He's here at his bar mitzvah, and he had hemophilia A, and he had an insertion of an L1 element into an exon of his factor VIII gene, disrupted this gene, and gave him the disease. And this was a truncated L1 element. The full-length element is 6 kilobases. This was about 3.8 kilobases. Had a lot of the signs of a retrotransposition event, long poly A tail at the end of the L1, and target site duplications of the endogenous exon 14 sequence. His mother, as you can see here, was not a carrier of the insertion. And so this insertion had occurred either in a germ cell of the mother or early in the development of this young man. We went ahead and we were lucky enough to isolate the precursor of the insertion, and after a number of different studies done by us and by others, particularly Jeff Buca and John Moran, we now know the structure of the full-length element. It contains an internal promoter that drives the transcription of the element. It contains a poor, relatively poor antisense promoter. There's an open reading frame 1, which encodes a nucleic acid chaperone protein, important for retrotransposition. And the open reading frame 2 encodes an endonuclease activity and a reverse transcriptase activity, along with a conserved region whose function is really not known at the present time. The 3' untranslated region ends in a poly A tail, and the whole thing is surrounded by short target site duplications. Here's the life cycle of the L1 element. It's transcribed. The RNA is exported to the cytoplasm where it's translated, and then after post-translational modifications, a substantial ribonucleoprotein particle is formed, and this particle contains ORF1 protein, ORF2 protein, and probably many other proteins and other RNAs. And we found recently that there are a number of RNAs associated with this particle which are going to go into processed pseudogenes. This particle needs to get back into the nucleus, and then target primed reverse transcription occurs. And here's what that's all about. The endonuclease of the L1 cleaves one of the strands, and the 3' hydroxyl at the end of this strand at the Nick site serves as a primer, and the L1 RNA serves as a template, and you reverse transcribe, and then by some hocus pocus that we really don't quite understand, you ligate, and presumably the reverse transcriptase actually makes the second strand DNA. but this is not well understood at the moment. Now, what's interesting about this kind of reverse transcription is it doesn't occur in the cytoplasm where HIV and other retroviruses reverse transcription would occur. Also, long-term or repeat retrotransposons are reverse transcribed in the cytoplasm. This is different. Reverse transcription is going on right on the chromosomal DNA itself. Very unusual. Okay, and this holds for essentially all non-long terminal repeat retrotransposons from all different species. Now, these elements have really had an important role in driving genome evolution. They can not only give you insertional mutagenesis, you can have deletions at the insertion site. Because the poly-A signal at the end of the L1 is really very weak, you can have read-through of transcription to the next poly-A signal, and then you can dump this whole thing, including the 3' flanking sequence, into a new site in the genome. You can have 5', what we call 5' transductions, where you can take a piece in front of the L1 and put it back into the genome in a retrotransposition event. And because these sequences are present in so many copies in the genome, you can have non-allelic homologous recombination where you have mispairing and crossing over, giving you deletions and duplications. And actually, I've just shown you 6 different ways these things alter the genome in evolution, but there are at least another 5 that I could show you on another slide. So there's a lot of ways that retrotransposition alters the genome during evolution. How about retrotransposition causing human disease? Well, it turns out now the number is up to 101. I showed you that first case. Now we're up to 101 cases. There are many more ALUs actually inserted causing disease than L1s. There are a sizable number of SVAs causing disease. 4 poly-A tracks where we don't know what the source of the poly-A is. We just know it's a retrotransposition event, but we don't know if it came from an L1 or an ALU because there's a lot of truncation in the retrotransposition process. And just in the last month, actually, a process pseudogene has been found that caused disease. Very unusual. Didn't expect that. It, again, was an X-linked disorder, a chronic granulomatous disease, and was discovered in a Dutch patient. So our best estimate on the frequency of these events causing mutation and potentially disease comes from 4,200 cases of neurofibromatosis 1, which is an autosomal dominant condition, and meaning only one hit is required, and all 4,200 cases were analyzed molecularly, okay? And of those 4,200 cases, of course, most were point mutations, but there were 18 retrotransposition events. So that's where I got the number of 1 in 250 cases, okay? So it's not that common a mechanism, but it does occur to some extent. Now back a while ago, John Moran in the lab worked out this cell culture-based assay for retrotransposition. And this is a very robust assay. Here's the element. He took, at that time, a neogene, and it was in a backwards orientation with its own promoter and its own poly-A signal. And it had been disrupted by an intron in the forward orientation, forward from this promoter. So splice donor, splice acceptor. And after transcription, this intron can then be removed, and you still have this neogene intact, but it's now backwards. But after it gets reverse transcribed and integrated into the genome, you can then have G418 resistant colonies occurring, and each one of these colonies will have at least one retrotransposition event. Now, more recently, it's been possible to switch out this neogene, put in a GFP gene, and do the assay quicker with GFP. And it turns out that disease-causing L1s are really highly active in this cell culture retrotransposition assay. How about the number of L1s that are active in the human genome as compared to the mouse genome? Well, we just have one subfamily of active elements, and within that subfamily, there aren't that many that are full-length and potentially active. And when you put them through a retrotransposition assay, you get about half of them that are active. And our estimated number now of active L1s in the diploid human genome is on the order of 100. 100 active elements out of some 500,000 of these elements, most of them truncated and dead, sitting in the genome. They make up almost 20% of our genome, but only 100 in each of us are active. In the mouse, in contrast, there are 3 separate subfamilies that are active, and you can see the estimate from analysis of the mouse genome is that there may be some 3,000 or so mouse elements that are active. So in the mouse, they actually account for a few percent of all mutations in the mouse. Most of the retrotransposition events in the mouse are due to IAP elements and other so-called endogenous retroviral elements, which are inactive in humans. Now, it turns out that over the last 8 years or so, surprisingly to me at least, there's been developing evidence for somatic insertions of L1 elements, not just occurring in the germline, but actually occurring in the somatic cells. And this was started by Mutri in the Gage lab and Kufal, who found somatic L1 insertion in neural precursors. There have been recent reports from Jeff Faulkner's lab and Chris Walsh's lab also about somatic L1 insertions in neural precursors. Somatic insertion, somatic mosaicism, has been found in mothers. There was one natural L1 insertion in a human patient where the mother of the patient was both a somatic and a germline mosaic. And also this recent processed pseudogene insertion was also a somatic mutation because the mother had about 15% of her white cells with the mutation. So she was a somatic mosaic. There's been L1 expression found in somatic tissues. You can transfect embryonic stem cells or iPS cells and see L1 retrotransposition. We and others have made transgenic animals and shown early embryonic insertions, very interestingly, with very little retrotransposition in germ cells, but lots in the early embryo, as though there's even more going on in the soma than in the germline. So a few years ago, Adam Ewing in the lab decided to see if he could find all L1s in any genome by a technique that he called L1-seq. And this depends upon there being a few nucleotides close to the 3' end of the L1, which are specific for human-specific L1s. And this is this AC nucleotide, only 100 base pairs away from the 3' end, and this G nucleotide, which is only 10 base pairs away from the 3' end. He starts out with an asymmetric PCR using just the 1 primer, so he enriches for fragments with the 3' L1, human-specific L1 sequence. Then he does 8 different reactions in parallel, each with a different anchor primer where there's a specific 5-mer at this end of the 3' end of the primer, and then an N5 sequence and a sequencing primer. So he runs a PCR between those primers, 8 different reactions. Then he takes those 8 reactions and he runs another set with the G-specific primer close to the 3' end of the element. Then he does a size selection, combines the reaction, does Illumina sequencing, and looks for regions of the genome where these sequences align to a potential non-reference L1. And that's shown here, where you can identify a non-reference L1 because these sequences, where there are different primer binding sites, and this one actually goes into a non-reference poly A, where there's no poly A here in the genome, and then you can prove this is a real L1 sitting there in that individual by simple PCR analysis. So here's a summary of some of the data that Adam got on human-specific L1 polymorphisms. There are now something like 2,300 different non-reference L1 insertions that have been detected in various data sets. And interestingly, any 2 individuals will differ by an average of around 300 L1 insertions. So Rusty and you and I, will differ by about 300. Now, on the other hand, yeah, probably, yeah. Well, but on the other hand, if you were the same ethnic group as I was, then we would have a lot more similarity in terms of our L1 sequences. Maybe we'd only differ by 100 or so then. But the estimate for the rate of L1 retrotransposition is now on average around 1 in 140 meioses. It's not that frequent. If you took all of the retrotransposition events, including ALUs, SVAs, L1s, it might be on the order of 1 in 25 to 30 meiotic events. Okay, so I said I was going to talk about cancer. Now, it's very interesting. Back in 1992, an L1 insertion was found into an exon of the adenomatous polyposis coli gene, which is a crucial tumor suppressor gene in colon cancer. And that was a somatic insertion because it was not found in the normal colon. Okay? Nothing was found over the succeeding 18 years. Okay, so we go to 2010, and Iskow et al. Used a genome-wide approach, a full genome sequencing approach of L1s, and found, this was in lung cancer, and they found 9 L1 insertions in 6 lung cancers, which were somatic. They were not in the normal tissue. And they studied 20 different cancers. So they had 14 lung cancers with 0 and 6 with these 9 insertions. In 2012, Lee et al. Found many L1 insertions in colon cancers and varying numbers in other tumors. Actually, essentially nothing in brain tumors like glioblastoma and nothing in leukemias and lymphomas. And our first study done by Sylvia Shoyam was in adenocarcinoma of the colon, and she looked at 16 tumors with 16 paired normal colons studied by this high throughput multiplexing technique. And she found over 70 insertions in, these were L1 insertions that were somatic insertions, validated by PCR and sequencing. And here is a chromosome wheel. Here's chromosome 1, 2, 3, et cetera, around to the X and Y. These blue here are the reference L1s, the ones that are in the database. And these orange are non-reference L1s that have been found in the various studies. And this internal group is the 70 or so insertions found in the colon cancers and not in normal cells from these individuals. So from the 16 tumors, there were many potential L1s found in the tumors with very high stringency, not in normal tissue, and 72 of these were confirmed by 3' end sequencing. Of these 72, 35 were also characterized at the 5' end. In normal tissue, none of 12 high stringency hits could be confirmed by standard PCR. In other words, there were 12 potential hits that were found in only normal tissue and not in the cancer, and they were looked at and could not confirm that they were present in the normal tissue. We also studied 3 tissues, cerebrum, liver, and testis from 2 cadavers, and found no somatic L1 insertions in these normal tissues. And here's just to show you the numbers of insertions in each of these individuals. There were 3 individuals that had no insertions, and here's one with 17 insertions. So there was a lot of variation. And we looked for factors that might correlate with insertion numbers. We looked at CpG methylation of line 1 promoters, and there were some samples with some hypomethylation, but there was no correlation with the number of insertions and the amount of hypomethylation. We looked at microsatellite instability, and again we saw some of the individuals that had microsatellite instability, but again there was no correlation with the numbers of insertions in the tumors. The only thing that correlated was patient age. And here you can see the age of the patient correlating somewhat with the numbers of insertions found in the sample. It's interesting, the somatic insertions in the colorectal tumors did differ somewhat from germline insertions, and we don't know the reason why this difference exists. Now, 27 of the 35 insertions that we could characterize had typical target site duplications, but a quarter of them lacked target site duplications and endonuclease cleavage sites, and this means that these were insertions that were endonuclease independent, meaning that they were probably filling in double-strand breaks in the DNA. And this kind of phenomenon has been shown to occur, but very, very rarely in genomic DNA. So this was very different to find a quarter of them showing up with the tumor DNA, where you'd expect well under 1% with germline DNA. All 17 of the insertions are truncated. The expectation would be for 30% of the insertions to be full length, but 0 were full length. And they were on average very short, 585 base pairs, which is shorter than expected. So these two are major differences between the insertions that are found in the germline, in the genome, and what we find in cancerous tissue. So we've done a second study now to look at the development of the insertions in the tumor process, looking at normal, polyp, cancer, and metastasis from 4 individuals, and found somatic events in precancerous polyps. And we now have similar findings from normal and inflammatory bowel disease. Inflammatory bowel disease is Crohn's disease, ulcerative colitis, and cancer in 5 patients. And other studies have found somatic retrotransposition in pancreatic and esophageal cancer. So the people that have done this work are Sylvia Shoyam and Tara Doucet, and a lot of the analysis was done by Adam Ewing. I should comment that Adam is now at the University of Queensland in Brisbane, and he's gone to Brisbane after doing his postdoc because he feels like he's got a better chance to get funding in Australia than he does in the US. I tried to get him to stay in the US, but it's amazing. He just felt like he had to go to Brisbane, and he's probably going to be right. He's probably going to get funded easily. And we'll see. And at Hopkins, I've had collaborations with Bert Vogelstein, Ken Kinsler, Steve Meltzer, Victor Valescu, and a pathologist that's been dissecting out these cancer forests, Laura Wood and Dongmei Jing. And we've had collaboration with Jeff Faulkner in Brisbane, and that's where Adam is now gone to work. Thank you very much. Yes, Joe.
2 questions.